Evolution is a fact. It's one of the most well-established theories in all of science, supported by a mountain of evidence from multiple fields of study. But despite this evidence, some still doubt or even deny the reality of evolution. As we'll see in this video, understanding the evidence for evolution is not complicated, and it's all around us, from our pets and our farms to our own bodies. First off, before watching the video, you should at least know that evolution by natural selection is the accepted theory of life's origins, implying that all life on Earth is related and shares a common ancestor. We won't explain it in detail here since we have an entire video on it, but in brief, all complex life, including us humans, evolved from a much simpler form of life over millions of years. How do we know this? Well, let's start with fossils. The fossil record is one of the most important sources of evidence for evolution. Fossils are preserved remains of organisms that lived in the past, and they provide a window into the history of life on Earth. By studying fossils, we can see how organisms have changed over time and how new species have emerged. Transitional fossils in particular are a crucial piece of evidence for evolution. If evolution is a slow process resulting in small changes over time, then there should be fossils to represent the transition between different species. These are fossils that show an intermediate stage between two different groups of organisms, providing a clear link between them. Well, such fossils exist. Archaeopteryx is a good example. It's a transitional fossil between dinosaurs and birds, with features of both groups. We even have sets of intermediate fossils, like this one, showing the progressive evolution of whales from their semi-aquatic mammal ancestors. Keep this in mind, we'll be referring to it later. Have you ever wondered why the majority of marsupials are found in Australia? This question can be answered using biogeography, which concerns the geographic distribution patterns of organisms and the factors causing those patterns. Biogeography provides evidence for evolution because it shows how the movement of Earth's tectonic plates and changes in climate have affected the distribution and evolution of different species. Around 200 million years ago, as the supercontinent Pangaea broke apart, populations of mammals became isolated from each other and they evolved separately, leading to the diverse array of mammalian species that we see today. As it turns out, the oldest marsupials are from the Americas, where they split from placental mammals at least 100 million years ago. Back then, South America and Australia were joined to Antarctica in giant landmass. At that time, Antarctica wouldn't have been covered in ice, but actually covered by a temperate rainforest climate. It seems that many marsupials and their relatives traveled from South America to Australia through Antarctica, and then became isolated when the continent broke apart during further continental drift. This idea is supported by the discovery of marsupial fossils on Seymour Island in the northern Antarctic peninsula. Island biogeography also provides evidence for evolution, as islands often have unique ecosystems and species that have evolved in isolation from the mainland. This isolation can lead to the evolution of new species through a process called speciation. There are two types of speciation: allopatric and sympatric speciation. We'll focus on allopatric speciation in this video since it's more relevant to biogeography. Allopatric speciation occurs when a population becomes geographically isolated from the rest of the species. The isolated population evolves independently until it's no longer able to breed with the original species, and a new species is formed. Darwin's finches on the Galapagos Island are a famous example of allopatric speciation. On each island that Darwin visited, he noticed the finches had different beak shapes that were adapted to the different types of food available on each of the islands. Embryonic development provides compelling evidence for evolution. For instance, all vertebrate embryos, including human embryos, have gill slits and tails. This occurs because all vertebrates share a common ancestor with ancient fish, in which these structures first evolved. Comparisons of anatomy outside the womb also support the theory of evolution. Anatomy is the internal and external structures of an organism. Vestigial structures are parts of the anatomy that don't serve an important purpose anymore, and are considered byproducts of evolution. The human appendix, tailbone, and wisdom teeth are commonly referred to as vestigial structures. These features once had a purpose in our ancestors, and as we evolved, they became obsolete but harmless, so they remained as part of our anatomy. Other examples of vestigial structures include the mammalian pelvis, present in modern whales, the tiny legs present on certain snakes, and the wings of flightless birds. In most cases, these wings aren't entirely useless, and evolution has adapted them to serve a purpose in running or swimming. Plenty of useful anatomical features are shared between species too. For example, homologous structures are structures in different species that are similar in form and function because they evolved from a common ancestor. Whether it's hands, wings, or flippers, upon inspection, all of these structures share a common blueprint that's adapted to serve a different function based on the organism's environment. Analogous structures are features of different species that are similar in function, but not necessarily in structure, and which do not derive from a common ancestor. How then do these structures provide evidence for evolution? Well, analogous structures illustrate what's known as convergent evolution. For example, both the Arctic fox and the Arctic hare evolved white fur despite not having a common ancestor with the trait. Clearly, white fur provided camouflage and it increased their respective chance of survival, leading to the selection of the trait. Analogous structures demonstrate that comparable selective pressures can lead to similar adaptations, despite the species not being closely related. In recent years, molecular biology has shown us that all living things share the same genetic material called DNA, the same or very similar genetic codes, and the same processes of gene expression, known as transcription and translation. This suggests that all life shares a common ancestor that had DNA as its genetic material. Using molecular biology, we can compare homologous genes which share sequences within or between species. An example of a homologous gene is the INS gene, which codes for the hormone insulin. This homologous gene is present in many different mammals, and it's important for the regulation of glucose levels in the blood. The INS gene is more closely related between humans and dogs compared to mice. This indicates that humans and dogs share a more recent common ancestor with respect to the INS gene than they do with mice. This relationship is reflective of the evolutionary history of these species. Despite these differences, insulin produced by one species can often function in another species. For example, human insulin can be used to treat diabetes in dogs, and insulin from pigs can be used to treat diabetes in humans. This is because the basic structure and function of insulin is conserved across different species, even though there may be a few genetic differences. Scientists can analyze homologous genes to actually compare how closely related different species are to each other. For instance, I asked you earlier to keep in mind the fact that whales evolved from semi-aquatic land mammals. Well, by comparing homologous gene sequences, scientists have found that the closest living relative of whales is actually the hippopotamus. In short, different organisms have the same genes, doing the same things, because they were inherited from a common ancestor. For some, it can be hard to fathom the process of evolution by natural selection, since it happens over such a vast period of time. But humans can accelerate the evolutionary process, and we've been doing it for thousands of years. Farmland animals such as cows and pigs have been selectively bred over generations to produce desired traits such as higher milk yield or leaner meat. Similarly, plants have been selectively bred for traits such as larger flowers or sweeter fruit. This process is known as artificial selection. It provides evidence for evolution because it shows how selection pressures lead to population changes over time, just as natural selection does. If you own a pet dog, one of the most striking examples of artificial selection is in your own home. In just a few thousand years, humans have been able to transform a single species of wolf into dozens of different dog breeds, which vary extensively in their shape, size, and adaptations. The vast changes seen in artificial selection demonstrate what is possible in just a few thousand years, making it easy to envision the potential of natural selection over millions of years. Darwin's theory of evolution provides a unifying framework for explaining the diversity of life on Earth. Everything you see in nature has been shaped by evolutionary processes, and evidence for evolution has never been more plentiful. Evolution is a story that's still unfolding. No doubt there will be new discoveries in the future that lead to greater understanding of our past and contribute to a profound sense of awe for the beauty and complexity of life.